Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Leah Rosen, online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. Because of the topic and volume of interest, today's Ask the Expert will run a little longer than the normal 15 minutes to allow for further discussion. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on our screen. After the presentation, we will begin the question and answer portion, because this is, after all, Ask the Expert, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you again for joining us today. It is now my pleasure to introduce our speaker, Chris Shields from Saint-Gobain Life Sciences Business Unit.
Thank you. Thank you everyone for attending this webinar. We're very excited to be presenting some of the data that we've been working on for the last 2 years. It's been a long process for us to bring some of this capability in-house and really to start evaluating our products and helping to lead the industry on next generation extractable programs. So as an overview of what I'm going to talk about today, I'll go through a brief introduction. We'll look at how Saint-Gobain does our extractable and leachable segmentation. We'll review very briefly some current industry guidance and standards that are either in place or being worked on. Then we'll look at the overview of all the Saint-Gobain single-use materials and products. We'll review some extractable tests. We'll look at the program development that we've done over the last few years. And then most of the time will be spent on looking at some variability testing that we've done on E&L, from analytical analysis to looking at the extraction methodology we've validated that really looks at our within lot variability. And then we'll do some Lotto Lot variability testing. We'll evaluate some post-treatments and then look at some temperature effects on some of our products. We'll follow that up with an overview of what we do for more complex single-use systems. And then we'll spend a little bit of time on what is an industry hot topic, and that's the BPOG extractable proposal and testing. And we'll give you some idea of how we're approaching that and how we're trying to meet the needs of both all end users, but specifically the BPOG group of end users. So if we look at how there's many different definitions for extractables and leachables. At Saint-Gobain, we characterize them into 4 categories. We have material characterization, and that's really the first step in the process of looking at a new single-use product. And in that step, we identify all the potential monomers, oligomers, additives, either that we know are intentionally added or may carry through from previous processes. Then the extraction step would be carried out under exaggerated but not extreme conditions, using model solvents at increased time and increased temperature with increased surface area to volume ratios. In addition, we have what we call in-process leachables, and that covers a very wide range of different test methodologies. So we're using really simulated process conditions where it could be it's generally an actual bioprocess fluid. We use the actual times in the process, the temperatures, and the actual process surface area to volume ratios. So this is more, like a bracketed leachable study. It's to provide end users with more information to do their risk analysis, but it's still not the final leachable testing. And so in our segmentation, the leachable testing is done with the final drug product and is performed by the drug manufacturer. So down below, I have a chart that kind of outlines an overview of where these different segmentations fall. So if you look at material characterization, it has the largest quantity of chemical compounds and also the largest number of unique chemical components. Then as you move into a subsegment of that of extractables, which are done in exaggerated conditions, and then generally a subset of that which is in-process leachables, which are done at conditions that are very similar to the actual process, and then the final leachable testing. If we look at where the industry stands today, there's a number of groups that have recommended and some standards organizations which have mandatory guidelines. BPSA has 2 solvents that they recommend, a water and a low molecular weight alcohol, and then 1 time and temperature. BPOG has 6 solvents that they mandate: water, a low and high pH, a surfactant, a salt, and a ethanol. And it's generally they have less than 30 minutes at 25 C, and then 24 hours, 21 days, and 70 days at 40 C. Now, there's a matrix of products that fall under these different times and temperatures, but generally for many of the products, it's either 3 or 4 time temperature conditions. So if we look at what the BPOG proposal is requesting for end users to provide, it's 6 solvents and 3 to 4 time temperature conditions, as well as post-gamma radiation and post-autoclave if both are used. So you could end up with anywhere from 24 to 48 test conditions per product. If we look at where the ASME BPE is, their current guidance that's going through for the 2016 standard, which still hasn't been approved, but it's in draft form, is they have 2 categories of testing. One is similar to our material characterization, where they recommend 2 solvents, 1 polar and 1 nonpolar. And then for their in-process testing or simulated process model solvents, they recommend up to 5: a water, high and low pH, a salt, and an alcohol. Generally, it's 40 degrees C for 30 days. ASTM E55 has a draft standard. It currently has 3 solvents in the mandatory and then 3 in a recommended. And the time temperature conditions are still to be determined. The PDA doesn't have any specific recommendations, but said extractable and leachable testing should be performed. And then USP 661.2, again, has very general language around extractables and leachables. If we look at the European Pharmacopoeia, EP 3.1.9, which is specific for silicone tubing, they recommend for one of their tests a water reflux at 4 hours and then a hexane reflux at 5 hours. So there's a lot of different solvents, different methodologies, times and temperatures that different groups have recommended. That presents a problem for a lot of the single-use manufacturers who have a large amount of different product types. And this chart shows for Saint-Gobain our single-use materials of construction snapshot, where we have products in several categories: tubing, hose, connectors, containers, manifolds, and systems. And I've highlighted the different materials that we use for those. For tubing, we have 6 or more materials. And then for each of those tubings, we may have multiple formulations. So C-Flex has 6 formulations. We have 3 additional TPEs, 6 additional silicones, polyethylene, polypropylene tubing. For silicone hose, there's multiple formulations. And then when we get into systems, we have multiple components, many of the components that are from other manufacturers. So it becomes very complex to achieve test results that meet all the multiple requirements out in the industry. In addition, we generally are doing our extractables as manufactured, post-gamma radiated, and then autoclaved. So overall, Saint-Gobain has over 50 products that we need to perform extractable testing on. So these products, if each product needs to have 20 to 40 different time, temperature, solvent points, it can become quite expensive. So part of what we've done for the last 2 years is really tried to understand how these different conditions impact specifically the Saint-Gobain materials of construction and product form factors, so that we can really have a scientifically based approach to our extractable testing. And we can really predict for each of these different standards how our product will perform, and if we need to do specific testing in that area, or if we have enough of a database of fundamental understanding of how the materials of construction and the product form factors impact to the extractables that we can make predictions. So for our extractable program, we're doing semi-quantification identification. We do testing to USP 661 and EP 3.1.9. More is a legacy test to have crossover to previous or older testing, so we can see if things are in the same general buckets of gross extractables. Then we do USP 643. Again, it's a good gross extractable test to really tell us if there's something different about the product or the testing. We do metals testing using ICP AES. We do volatile organic compounds using Headspace GCMS and Headspace GCFID. We do the semi-volatiles using Direct Inject GCMS. And then we do the non-volatiles with LCUVMS or LCMS DAD. So we started this program in 2013. And really that was the point that we started looking at all of our products and trying to characterize them from a more fundamental understanding of how the polymers are compounded, how they're extruded, what materials go into them, what additives there are, what the potential extractables could be. So we spent several months compiling these lists of potential extractables. During the same period of time, we were identifying, qualifying, and validating some analytical equipment in our research center in Shanghai. We had a significant analytical operation there that was supporting microelectronics. So we were able to really tap into their ultra-clean product analysis capabilities there. In 2014 and 15, we really focused on developing, qualifying, validating different extraction methods and trying to come up with a standard that we felt had the lowest level of variability. Then we focused in this year on determining product and process variability, really lot to lot, within lot, and within the analytical equipment. Through the second half of this year and through next year, we will complete our existing backlog of product testing using our standard test format. Next year through the beginning of 2017, we're going to be focused on doing some experimental designs that will really map out how our materials of construction and product form factors react to different solvents, different times of exposure, different temperatures, and different post-treatments. And then during that same time period, we will complete BPOG testing on a number of our products. So this slide really is just kind of a snapshot of some reproducibility testing we've done. And this was testing done on the same extraction. So we extracted, in this case, some polypropylene connectors with water and ethanol. We took that extract and we did the analysis through our analytical equipment 3 times. So if we look at this chart here, the water VOC chart, we can see that there are 3 different injections, and we had the equivalent amount of concentration of all the cumulative extractables. So we found in this one that for each of the injections, we had the same level. So there were 2 compounds identified, and they had the same overall concentration. No extractables were detected for SVOCs. For NVOCs, again, we had one chemical identified with the 3 injections, and they had relatively the same amount of concentration. In ethanol, we had no extractables detected with VOC analysis, but for SVOCs and NVOCs, we had quite a few different extractables. On the next slide, I'm going to focus in on these 2 slides and give a little more detail. So if we look at the SVOCs using our direct inject GCMS, you can see that we have a number of compounds identified. But if you look at the levels at each of the injections, they're very, very consistent. Here we're in micrograms per centimeter squared. So these are all in the, these tests were run on the connectors at 3 centimeters squared per mil. So these are all generally in the range of just around or above 1 ppm. So you can see that some of these compounds are detected really at our limit of quantification, which is down below 500 ppb. So you can see that in each of these chemicals, we have very little difference between each of the injections. So overall, our analytical equipment on our GCMS has very good reproducibility from injection to injection. If we look at the next one, which is our connectors for non-volatiles using LCMS UV, we can see again that we have 8 different compounds that were identified. And generally, we have the same concentration in each of the compounds. When we get to this last one, the injection, we only detected it 1 out of 3 times. But again, at these levels, we're down at our level of quantification and our level of detection. So it's not surprising that we may or may not see the peaks. This is a summary of all of that data. So for VOC analysis, with the water injection, we identified 2 compounds. The cumulative concentration was 0.26 micrograms per centimeter squared, the same across the board. Nothing detected in SVOCs. NVOCs, again, very consistent results from 1.03 to 1.18. For the ethanol testing, again, nothing. For VOCs, 13 compounds detected for SVOCs, very consistent results. NVOCs, we saw either 7 or 8 compounds and very consistent results. If we move to within lot variability, this is the same testing. So this is one, for these we had a connector, a tubing, and a bag. And for each of these tests, we took the same lot of material and did 3 different extractions on it. So this is really showing as closely as we can manufacture how our extraction method varies. And again, you can see there's very little variability from lot 1.1 to lot 1.2, which is the same lot, just extracted twice. A little bit more variability, but very small compared to the overall levels. And again, very little variability from our one extraction to the next. Again, I'll focus in on one of them specifically so we can get a better idea of that, but we had 3 different extractions on the same lot of material. And again, we saw very consistent results from all 3 compounds that were identified. In summary on that, for the connectors, 2 compounds identified, overall very similar VOC numbers. 1 compound for the NOCs, for the NVOCs, very consistent. So very consistent results across the board from within lot variability, which is really assessing our ability to do an extraction reproducibly over time. This is the same within lot variability on ethanol. Now, as you can see from this chart, ethanol pulls out a lot more compounds than water does. And we can see, though, that there's still very consistent results, even though we're pulling out a lot more compounds. I'll focus in on one of the specific ones. So for tubing NVOCs, using LCMS UV, we can see that we've identified quite a few different compounds, but each compound is very consistent from extraction to extraction. So we have a very consistent extraction process. Overall, the summary of that, I'll just focus on a few of these. So even when we get to the very high levels of compounds where we're seeing 15, 16, 17 compounds, overall we have very consistent results from extraction to extraction. Testing reproducibility. This is lot to lot. And again, so we've tested multiple lots and looked at the variability between lots. And again, for tubing, VOCs, very little came out with water, but it was very consistent what did. For bags, again, very little came out with the water, but very consistent between lots. The same thing with the NVOCs in the bags. Focus in on one of those. The bag NVOCs, again, from chemical compound to chemical compound, very consistent. Again, here we're again down at the levels of our quantification and detection, so it's not surprising that we didn't detect repeatedly one of the compounds. Again, if we look at lot to lot, in summary, very consistent results from lot to lot, from VOCs all the way to NVOCs. Here, this is looking at, again, with ethanol. I'm going to focus in on this next slide. So here we're looking at the post-treatment effects of connectors. And these were done at 70 degrees C, 24 hours in water. And if we look at a post-autoclave versus a post-gamma radiated, we can see that we generally get more out after post-gamma radiation. And that's been our consistent finding we've had across the board for all materials of construction and product form factors. Again, if we look at temperature effects, here we're testing connectors, and we have it at water at 25 degrees C, water at 70 degrees C, ethanol at 25 degrees C, and ethanol at 70 degrees C. All of them extracted for 20 to 24 hours. For the lower temperatures, we don't extract anything. When we go up higher in temperature with water, we do see some NVOCs in the connectors. Ethanol at low temperature, we see no extractables except at the NVOCs. And then as we get up into higher temperatures, we start to see more extractables on the NVOCs. So overall, we've found that our variability of our analytical equipment from injection to injection is quite low. The variability of our extraction method is very reproducible. There is a little bit of lot-to-lot variability, but generally it's small compared to variability we've seen in other things like the difference between autoclave and gamma radiation or lab-to-lab testing variability. So if we look at how we test large single-use systems, this is just a snapshot of 3 products from the same family. And it's a grouping of bottle manifolds with sterile connectors of different sizes. This one's a 1-liter bottle bag sampling system. This one's a 2-liter and then a 10-liter. We find it challenging, and customers request that we get validation information, including extractables on these systems. Many of these systems have components that don't have full validation data from other suppliers. So what we've done is we've started putting together monster systems where we'll take a family of products and then do full validation testing, including E&L, where we can either do a composite test at a higher surface area to volume ratio, or we can do it more of in-process leachable tests where we do the actual conditions. The last topic I want to cover is the BPOG extractable recommendations, which is an ongoing effort for us. We're looking at providing support to BPOG and other end users for tubing, bags, and connectors. On tubing, we'll be selecting certain products that will be tested according to the BPOG proposal, some silicone tubing, some silicone tubing, some C-Flex, some other TPs. bioprocess bags, pure fit SIB. And generally, it'll probably be post-gamma radiation since we've found that to be worst case. But our timeline for this is, the first half of 2016, we're doing a DOE focused on materials of construction, product form factors, solvents, times, and temperatures. We'll then follow that up with a survey to the BPOG members on key products that they want us to test to the full protocol. We'll present our scientific support for BPOG, for the BPOG proposal, and then we'll complete the BPOG testing towards the tail end of 2016, the beginning of 2017. So that's the end of the presentation and the end of my half hour of time. And I'd like to thank the Saint-Gobain Extractables team who has put a tremendous amount of work into this effort over the last several years. And it's an ongoing effort for us and has a very high level of support within our organization. Thank you.
Thanks, Chris. So our first question is regarding the extractables and leachables in the C-Flex tubings, is the antioxidant LRGAFOS 168 used as a raw material?
Ergophos 168 is in almost all polyolefin materials that are processed. So where C-Flex does have polyolefins in it, it is one of the components that is extracted out.
And then do you identify all the compounds detected, I think referring to slide 20 and 22?
So in general, we've been able to identify almost all, we've been able to identify all of the VOCs and almost all of the SVOCs. We have just a couple more across our product line to identify. We have a few more that we need to identify with ethanol extraction for the NVOCs, and that's an ongoing effort for us. We have a major program in place for 2016 to work with some outside labs who have advanced analytical capabilities to help us to identify those compounds. But our goal is to identify using all the available means that we can, all of the compounds above 1 ppm, and then we will make best efforts to identify everything from 500 ppb up to 1 ppm.
Okay, and what is the ratio you selected for the bag assembly extraction?
So the bags were done at 6 centimeters squared per mil. So again, the bags weren't fully filled. And we've tested both 3D and 2D bags. We like to test the 2D bags because they give us, it's easier to get higher surface area to volume ratios.
Okay, so this next question is a bit long, so I'm going to break it down. But let me know if you need a reminder of what the question was. So can you say more about what you do with results, and do you have acceptance criteria? How much do you work with a customer to perform risk assessments to determine product-specific acceptable levels?
Yep, so this information is available in a couple of different venues. Our standard test methodology will go into our validation guides, which is available online. You have to register on our site and then be approved to access certain documents. But sometime by the end of the first quarter of 2016, all of this data for almost all of the products will be in our validation guides online. The more detailed information that isn't just a standard extractable test, we're going to be publishing over the next few months several white papers that again will likely be available on our online site.
Okay, and which tubing size?
I missed the last part. So how do we determine specific acceptable levels? Right now for the our extractable testing, it's more of a pre-product assessment. Are there any chemicals that are a concern? Are any of the levels extremely high that may be a concern that should have further analysis done? We really get into doing more acceptable levels and doing more detailed risk assessments, more on the in-process leachable testing, where we work specifically with the customer on process-specific conditions and generate the data. So those are the ones we spend a lot more time individually with customers doing risk assessment.
Okay, so how cooperative were your resin suppliers with the composition information?
It's interesting because we expected our resin suppliers to have a high level of knowledge of their products. And in some cases they do. I think our silicone suppliers are very good in that. The polyolefin suppliers tend to be, I think, less knowledgeable. We even had one instance where the supplier told us one of our unknowns wasn't in their resin. And we actually then had to test the resin, and we found that it was in the resin. So it's a mixed bag on how helpful the resin suppliers are.
So here's a question about the BPOG. Do you plan to do all points that BPOG requests, or will you try to use a rationale to limit the working cost?
So our plan is to really develop a scientific basis for understanding each of the materials of construction. There are some products that, it doesn't make sense to test all the BPOG conditions. For instance, our FEP PharmaFloor tubing, very little is extracted out of it under any condition. So it wouldn't really be prudent or cost effective or generate valuable data for our customers to do a lot of testing on PharmaFloor. There are some other products that may be sensitive to different pHs and different time points, and so those products would be more likely to be tested to either a partial or a full BPOG proposal. But our plan is really to work with the BPOG members and understand which of our products are key to them and need to have the full BPOG proposal, whether or not supported by scientific data that we've generated. So that's part of what we're going to do. We'll do a survey to the BPOG members and to the industry at large to find out, is there one C-Flex, one silicone tubing, one of our TPE tubings, really that is the one we should be doing the full BPOG proposal to.
Good. And how do you address lot-to-lot variability in extraction results?
It's an interesting question because I think this is the first time we've done a real systematic evaluation of it. And, it made us nervous when we first started looking at it because we didn't know what the results were going to be. I think we're pleased that the results are as consistent as they are. I think we have more work to understand our raw material variability. But I think because this testing really is, semi-quantitative, and we're not using, standards for every peak that is generated, I think the variability of the product is within that variability of a semi-quantitative method.
Okay, well, thank you very much, Chris, and thank you all for attending. Again, this presentation will be housed in the multimedia section of the Bioprocess International website. You'll be getting a direct link in your email, so watch your email if you'd like to play it again on demand or share it with colleagues who may be interested. Thank you so much for joining us.
Thank you, everyone. Bye.